This is a case study involving genetically modified tomatoes, specifically tomatoes that are genetically modified using novel organisms. The tomato is an economically important crop due to its wide consumption as a vegetable and its use in a processed form in the food industry. The U.S. is a world leader in the production of fresh tomatoes. The tomatoes are host to a wide variety of pests, including aphids and lepidopterans. Control of these pests is now done through chemical insecticides, but many of these pests are developing resistance. Therefore, genetic modification provides an opportunity to develop organisms that might be resistant to pests and might require less chemical pesticides. For this case study, two species will be contributing to the genetically modified tomato. That is the snowdrop and the Australian funnel web spider. From the start, there is something of a balancing act this GM crop will have to navigate. The first is that it has to be safe or relatively non-toxic to bees. And the second is that it has to have specific properties against lepidopterans and aphids, pest species. Genes from two separate organisms were selected. There's an agglutinin from the snowdrop, from a plant, and there's spider-venom. The agglutinin was chosen because it is a lectin. It is a carbohydrate binding protein. It is found in plants known to act against sucking and chewing insects. Ingestion can lead to the disruption of essential physiological processes, leading even to mortality. The snowdrop lectin has specific toxic effects against hemipterans. The spider venom was chosen because it is a calcium channel blocker in the insect central nervous system. It is lethal to a broad range of insects, but studies show it is likely honeybee safe. Importantly, it is not known to cause inhibition to mammalian voltage-gated calcium channel currents. The peptide is of limited oral toxicity to insects, but fusion to GNA means it's a carrier molecule to insect hemolymph of target insects. From the start, it's important to realize that this novel GMO will be incorporating spider genes to encode for venom. The spider venom genes may encode proteins or peptides with specific toxicological properties unfamiliar in agricultural food systems. Spider venom has been used as a GMO in some organisms against mosquitoes, but the potential to harm non-target organisms, NTOs, like pollinators, predators, and decomposers, must be evaluated rigorously. Spider venom toxins are often highly specific to certain taxa, certain insects or arthropods, and assessments will need to determine whether those genes affect only the intended pest species or if they inadvertently harm beneficial insects or NTOs. Proteins or peptides expressed by venom genes may have different degradation rates in the environment, particularly affecting soil microorganisms, aquatic systems, or broader ecosystems. So taken together, this potential GMO is not like BT crops at all. When using a novel GMO, it's important to consider that there can be unintended effects on food chains. This might lead to cascading effects in the whole ecosystem. This ripple effect must be modeled in an ecological context. There are also regulatory considerations. using genes from non-food sources introduces a higher burden of proof for safety since these genes are less familiar. Regarding non-target organism assessments, there can be a tiered approach. The first tier would be laboratory-based testing. Stage 1 of this tier would be to search databases for venom proteins and other toxins and allergens that could have structural or functional similarities with the venom protein. Next, there could be toxicity testing on key non-target organisms, ranging from honeybees and ladybugs all the way through aquatic invertebrates. Next, quantifying the expression levels of the venom protein in the plant or organism and focusing on tissues likely to be consumed or contacted by the non-target organisms. And finally, degradation and persistence studies. Tier 2 involves semi-field studies. In other words, an enclosed area of a field, perhaps with netting, perhaps in a large greenhouse. In these enclosed field trials, there'll be an evaluation of the interaction between the GMO and the NTO. Particular attention can be paid to behavioral studies, assessing changes in foraging, reproduction, or survival rates for things like beneficial insects, pollinators, and soil organisms. Tier 3, the next and final step, is the long-term ecosystem monitoring in the field. This is monitoring NTO populations and ecosystem balance over extended periods in areas where the GMO is deployed. This leads to a landscape level impact assessment where the GMO's broader impact on biodiversity, including non-target arthropods, birds, and mammals. The tiered structure of analyzing a potential genetically modified crop clearly relates to BT crops. The first commercially available genetically modified crop was the flavor saver tomato, but the crops that captured the majority of the attention were the BT crops. BT proteins likely target a narrow range of pests, while the spider venom proteins may have broader or unknown effects. BT proteins primarily act on pests after ingestion, whereas spider venom proteins have other modes of action, including contact toxicity, in addition to ingestion. This requires different exposure models. There are novel exposure pathways, and spider venom proteins may persist differently in the environment compared to BT proteins. Genes sourced from non-food organisms like spiders may face heightened regulatory scrutiny. This requires a totally different baseline of data, and venom-derived proteins require new baselines. There are a variety of non-target organisms that could be sampled because they will be impacted by this GMO. These could be things that would be located far away from a field, like in an aquatic environment. There are also different exposure pathways that might be involved, and specifically with regulatory agencies like the EPA, there could be a need to include standard indicator species when devising an experimental approach. There are a variety of potential exposure pathways, and NTO might encounter the GMO protein by ingestion of plant material, by contact with environmental residues, or if it's a predator, by consumption of prey that has bioaccumulated the protein. It can be absorbed into the NTO's system in a variety of ways, and it could have an acute toxic effect, a immediate death or significant physiological disruption, or it could have sublethal effects over a period of time. A non-target organism must be susceptible to the protein. This includes receptor compatibility, the likelihood of exposure. It has to live or operate in environments where the protein is present. It must lack specific protective mechanisms, and more importance will be given to a non-target organism that plays a critical role, like pollination. Key pieces of information are needed to test various pathways. That's environmental exposure, the physiological susceptibility, the toxicity data, the ecological role and interactions, and the protein degradation. Here is a chart that outlines specifically the key pieces of information that are needed in order to test the pathways. Different NTOs need different kinds of tests to determine their susceptibility. This includes specificity testing, dose response studies, as well as protein selectivity. There are special regulatory implications for a new genetically modified organism. The EPA, the FDA, and the USDA are all involved in regulation. The Coordinated Framework for the Regulation of Biotechnology, established in 1986, describes how the agencies work together to regulate GMOs. This would likely involve an extensive period of monitoring post-deployment. This would be long-term field studies may be necessary in order to monitor actual impacts on NTO populations and ecosystems. Risk assessment will have to be extensive. This includes broad testing requirements, a detailed analysis of indirect effects, specificity analysis on protein target interactions to minimize off-target effects, and regulatory oversight. There are challenges and safety concerns such as non-target effects, including on other plants, beneficial insects, and higher organisms. Human health, of course, was just touched upon here, but will be a major focus. And long-term, the issue of resistance development is important. These are some of the references used in the presentation. Thank you for your attention.